Welcome to Shellphone, the podcast that gives the ocean its very own hotline. Join us as we hear from ocean stewards, discover threats to ocean health, and learn ways we can all answer the ocean's call. This podcast is brought to you by Breach the Surface and Coastal Creative. I'm Harmony Dawson, and joining us this episode is John Parkinson, who's a professor at the University of South Florida. Hi, thanks for joining us today.
Thank you for having me. Happy to be here.
Awesome. I'm super excited to talk about corals. I've been wanting to talk about this topic for a long time. But before we get into that, can you give us a little introduction into who you are and what the work that you do is?
Sure. So I'm John Parkinson, an assistant professor at the University of South Florida in the integrative biology department. And my lab is really focused on coral interactions, those between the host and the symbiote. And we'll talk about how corals are really excellent examples of symbiotic organisms. And so we use molecular methods. We use a lot of DNA sequencing and a lot of DNA sequencing and microscopy and some other techniques. And then we go out into the field, we collect corals, we do experiments on them, and then we also work on a model for corals, sea anemones, in our lab. And we use those tools to try to understand corals, what makes them tick, how partners recognize each other, interact with each other under both normal conditions and stressful ones, and how coral bleaching happens when you have a stressful condition. So the whole underlying goal ultimately is to know more about corals and how they work so that we can apply some of that information to coral restoration and conservation efforts.
Awesome. That's a great overview of all the topics that I want to touch on today. So hopefully we're able to get to all of that because there is a lot to talk about. But before we do that, I just want to start with a general overview of what corals are and like what the major important points are to know about them. So what makes a coral is my first question.
Well, that's a good question. Sometimes we have a hard time defining what a coral is or what an individual is when it comes to corals. Corals are animals, okay? They're closely related to jellyfish, but instead of swimming through the ocean their whole lives like jellyfish do, they end up stuck on the ocean floor, and they actually grow their own skeletons that cement them to one particular place, and that's where they spend their lives. They are clonal. So when you see a coral colony, what you're seeing is this superorganism that includes a lot of different individual polyps that are all connected to each other, and they bud off of each other, and that's how these corals grow. So if I went down and I sampled one little polyp off of a coral, and I stuck it on a petri dish, technically it could survive entirely on its own without any of its clonal brethren. And so if you take a coral colony and you fragment it into multiple pieces, you now have multiple corals, although they're all genetically identical to each other. Corals are not just animals in the sense that they also associate with other organisms, a lot of microbes. The most important microbe they associate with, in my opinion, is algae. And these aren't the big macroalgae that you see growing like kelp forests, right? These are microalgae. They're unicellular, and they actually live inside of the cells of the coral host. And they can photosynthesize like a plant, and they transfer that energy that they produce to the coral, which allows it to lay down its skeleton. And so a coral colony will actually grow over time by laying more skeleton and providing more area for those polyps to cover. Because the living coral is actually just this very thin tissue layer on top of the skeleton. So if you're walking along the beach and you pick up a piece of coral, usually what you're picking up is a piece of the skeleton. It doesn't have any live tissue on it anymore. But if you go out into the water and go snorkeling or scuba diving, you'll see these, beautiful multicolored colonies. And the reason that they are colorful like that is, one, they have a coral tissue on top of it, and for the other, they've got those algae inside of that coral tissue, and that makes them cool browns and greens. But of course, the coral gets stressed and it loses its algae. They bleach, and we call it bleaching because they turn white. You can see straight through the host tissue to the skeleton underneath. What else about corals? So it's not just microalgae that they associate. They've got viruses. Fungi even live in there, a bunch of crazy things. So it's really this sort of community of organisms, but the coral itself is kind of the structure that holds all of that together. And it provides habitat for a bunch of other organisms.
Awesome. You've already touched on a lot of the different questions that I wanted to ask about generally what corals are and what's the deal with them. One question that I had is how you kind of define like what an individual coral is, what the colony is, what like a group of different corals would be.
Yes, those are all, again, those are difficult things to do. I mean, you can't just go out, swim up to a coral and say, hey, are you related to him?
Yeah.
Right? Or are you even identical to him? And that's one of the reasons we use a lot of molecular techniques in this field now, because if you want to tell individuals apart, you have to look at their DNA. Another interesting thing that comes with that is you can't look at a coral and tell how old it is. You'd imagine as time goes on, corals get bigger, which is true. If you see a very large colony out there, it's probably really old. And depending on what type of colony or which species you're looking at, it could be decades or even hundreds of years old, potentially. So for a big coral, great. But those smaller corals, well, are they just a fragment of that big older coral, right? We can't really know how old a coral is just by looking at its size. And actually, even some big corals, maybe it was an old big colony and it died, and then a new coral came and grew just over top of it really recently. So it's very hard to actually tell how old a coral colony is unless you're watching it from birth, which we sometimes do in the conservation space, which is one way that we know that many corals aren't actually reproductive until they reach about 4 to 5 years of age. So if you want to restore the species and you want these corals to be self-sustaining and reproductive on their own, coral that we plant today might not spawn again until 4 years from now, which makes it a challenge, right? These are long-lived organisms. And that's one of the reasons we use somewhat shorter-lived organisms in our lab to answer questions that we can't wait for out in the field.
This is kind of another silly follow-up to that question. If you were talking about like a coral reef, would that technically be like a colony of colonies of corals, or is that just totally silly?
I like your idea. That's very chaos theory. No, so the fractal. Coral colony itself is one entity. We sort of define colonies as physiological units. So if they're physically separated from each other, those are two separate colonies, whether they are clonal or not. What a reef is actually a community of multiple organisms and multiple corals, right? You can find corals that don't create reefs. If you go out deeper, right, you just get one coral colony. One colony a reef does not make. You have to have long stretches of many colonies together. And we find those in unique places in the tropical shallow marine ecosystems. And the whole reason that corals can be successful in those shallow, very low nutrient waters is because they have their association with algae. That's the whole benefit of the association, really. This was habitat that the corals couldn't really thrive in on their own, because corals can actually catch and eat their own food, right? They have stinging cells, or I mentioned it's like a jellyfish. So same group overall, and they all have stinging cells. They capture small prey items and consume them, but that's just a relatively small part of their diet. It's an important part because they can get nutrition that way that they don't get from the symbionts, but overall, they need the energy that comes from the algae to have the capacity to calcify and lay down skeleton in those shallow, low nutrient environments. And so once they were able to associate with these algae, this whole area that they weren't successful in before became something that they could be successful in. And many species diverged in those habitats, and that led to a lot of the diversity we see today. But yeah, if you go diving on a coral reef, you're going to see many different species living together, sometimes happily, sometimes they're in constant conflict with each other. They try to grow over each other. They actually send out extra special stinging tentacles to fight off. There's a lot of competition for space, just like in a rainforest, right? And so there are many different scales. You can go to one individual polyp, or even within that polyp, you can look at, different symbionts that might be in that one polyp. You zoom out, there are multiple polyps. You zoom out, those polyps are all on one colony. You zoom out from that, there are multiple colonies of one species. You zoom out from that, there are multiple colonies of different species. And together they're forming this large structure that you can actually see from space in the case of the Great Barrier Reef, for example.
Yeah. I know reefs are kind of the major thing that people think about when they think about corals. Well, tropical reefs, I should say, are what people really associate corals with. In a recent episode, we mentioned briefly deep water corals and all the different types that you might not first think of. So there's definitely a lot more diversity, even beyond the diversity that you see in tropical coral reefs.
Yeah, and yeah, those mesophotic deep reef ecosystems, some of them are in the tropics still, right? And we're finding new things all the time. Even in Florida, we randomly, someone goes diving somewhere people haven't been in a while and go, oh man, look at all of these acropora that we didn't know was here. Stuff like that happens. But I'm thinking also about down in Colombia, I believe, within the past 5 or 6 years, they discovered a whole reef that they didn't know was there because the water was so turbid, which means it's silty, it's junky, it's got a lot of particles in it, and so they couldn't see them. But if you go under that layer, there was this whole reefscape there that was apparently doing okay because enough light was getting through to them. But yeah, if you want to talk about reef diversity, I mean, there are reefs that are made by things other than corals. They're not coral reef ecosystems. They're other types of reef ecosystems. Oyster beds are a good example. You can have oyster-based reefs. So any living organism that can calcify and produce substrate that things can live on can technically produce a reef, I think, if it gets large enough. Don't quote me on that. Again, I'm not the geologist. They would know. But yeah, there's tremendous diversity in these ecosystems.
That's super cool.
Just at the coral level. And then you expand it to all the organisms that live on it. It's crazy.
Yeah. A quick side note on the reef that you mentioned that was kind of recently discovered, you could say. In preparation for this episode, I was listening to an episode of another podcast that I usually always go to when I can find an episode that relates to a topic that we're doing, and that's the Ologies podcast. And they had an episode on corals, luckily. So I was listening to that, and they mentioned something about how sediment is like an enemy to coral growth because it blocks out light and they just don't really jive together. So that's really interesting that was brought up here because I wouldn't have even thought of that before.
Oh yeah, the extra danger that comes from silty water is that it can make it hard for corals to find substrate. So when I say substrate, I mean a surface that they can settle on. And so I'm talking about how these corals are stuck in one place for their lives. That's not true. Right at the beginning, when they're babies, they're just these little larvae. They are dispersed into the water column and they flow where the currents take them. And they cross their fingers that they end up by reef habitat or something reef-like that they could settle in. And there are cues that, draw them there and attract them there. But you want a nice clean surface for them to lie on. And if you have too much sediment on a reef, not all of it is up in the water. Some of it comes down on top of the surfaces. And the larvae can't really settle on something that's covered in dirt, right? It would have to dig to get down to lay a foundation where it can start forming its skeleton and then grow from. And even just a little bit of silt can throw things off. So there are all sorts of dangers to corals in their first steps of life that I'm sure we can get to. But yeah, that was a cool discovery. And I should mention maybe that, I say it was discovered by scientists. It's entirely possible that the locals knew about it beforehand, right? A lot of times I'll also talk about probably how we've been describing new species of the algae. It's not that they haven't been there the whole time, right? They're sort of new to science and new to being part of that conversation and available to study. But you also got to recognize a lot of these places people have known about for a long time, but the word hadn't gotten out yet.
This is not a question that I was planning on asking before, but it just popped into my head. You mentioned that when corals are first babies, they're like larvae and they're kind of carried around by currents. Do they still have their own like free movement, or do they just fully go where the water moves them?
Well, it's going to be a challenge for something this big to fight the ocean, right? But they do have a surface that's coated in cilia. So it's, again, a question of scale, right? They are able to swim down, but they're sort of at the mercy of the currents to bring them to places that might work. And then, if conditions are acceptable enough and the water motion brings them near to the benthos, the lower surfaces that they want to get to, then they do have capacity to sort of self-propel. And once they get there, they have this seeking behavior, and they will try different spots, and they'll maybe start to settle down one and go, oh, no, that's wrong, pop back out and find another one. They like to hide from the light early on, so they usually find little cracks and things to try to swim into. Yeah, the whole life cycle of corals is pretty cool. They're towards the base of the animal tree of life, right? And so they've got all of the possible ways of reproducing that you could imagine. Some coral species have male colonies and female colonies. Some are strictly hermaphrodites, so they release both male and female gametes into the water. Some are serial hermaphrodites, so they'll start off as male and then later they'll become female. And then in terms of how they actually reproduce, some spawn, so they release those gametes straight out into the water, and all of the fertilization happens at the surface because they're sort of positively buoyant. They float up to the top and then they mingle with each other, and then those larvae are carried away. Some are brooders, which means that they retain the eggs inside, and so another coral will release sperm and fertilize those eggs, and then the grown larvae will be released instead of eggs from that colony. And these spawning events are really cool. I don't know if you ever had a chance to go snorkeling or scuba diving during a spawning event. It's you try not to think too hard about what you're swimming through, but it's actually really, really cool. It usually happens at night. And so in addition to it being a night dive, everybody on the reef knows that these corals are spawning. There's love in the air, essentially. Everybody knows it. And these little packets of sperm and egg make delectable bundles. There's a lot of energy there. So everybody comes out and tries to eat them. And so that's what part of the strategy of releasing so many of them on the corals part is, 99% of them will not make it to be adult corals, but the few that do could stick around for a very long time. And yeah, spawning, for many species, it only happens once a year. And it's like clockwork. Full moon in August, 3 nights later, 10 p.m. to 11 p.m., that's when the corals are going to spawn. And it used to be very easily predictable. It's less so now because so many of our corals have been disappearing, but that's another topic. But yeah, we can they're cued into the lunar cycle, and changes in temperature, and these things help them sort of coalesce around a certain night, and they all tend to go together. And in places that have multiple coral species, sometimes many of them all go at the same time. Multiple species, we call it a mass spawning. They have these in Australia all the time. Well, every year. And you get these giant slicks of coral babies, essentially, that then go out and do their own thing afterwards. So yeah, corals can both bud and clone themselves, and they can also reproduce sexually. So they can do it all. It's impressive.
This might be another silly question. Is there a way that corals communicate with each other? You said that they kind of figure out based on the conditions and the timing when to all kind of spawn around the same time. Mm-hmm. But outside of that, do they have any methods of communication with each other? I
think if Jack the colony is late or doesn't cue into the same lunar cycle on a given night, he might also recognize through chemical cues that other corals are spawning nearby and go, oh, it was tonight. I don't want to miss the party. So yeah, they definitely can convey signals to each other chemically. They can detect changes in the environment. They're light sensitive, for example. The sea anemones that we work with that are closely related to corals, they will retract if you have if you pass your hand over them and there's a shadow. Right? And corals will do the same thing. They also respond to touch. So if you don't do this, if you rub your finger across a coral, I can do it because I'm a scientist. I have permits for it. No, but you don't want to abuse or touch these guys in the wild. They're sensitive, and it's best to appreciate them from a distance. But if one did rub a finger against a coral, they would retract at those areas, and they might actually trigger the rest of the colony to retract too, because even within that clonal colony, there's signaling that's happening. I won't pretend that we have all of the signaling figured out in these organisms, but they definitely have means of communicating with each other.
Awesome. Then before we go into talking about your work specifically and also conservation in general, going back to the basics a little bit on the topic of the symbiotic relationship that corals and algae have with each other, what does it mean to have a symbiotic relationship?
So a symbiosis, and I should be able to explain this because I teach a symbiosis course, right? Symbiosis is just the living together of two different species for a prolonged period of time when they're in close contact. So a symbiosis doesn't necessarily have to mean that you have one organism living inside of another like we do with corals. A good example is sea anemones and clownfish, right? Nemo likes to hide out in his sea anemone. They're both benefiting from that relationship. Symbioses aren't always beneficial to both partners either, right? You can have parasitisms that would count as a symbiosis. So really, we're focused on mutualistic interactions, and in particular, in my field, we're interested in mutualistic associations with microbes, and this has gained a lot of popularity recently, not just because of corals, but because there's been so much interest in the human gut microbiota, right, and how bacteria help us break down and absorb our food and our diets, and how important a role they may play in our overall health. And when it comes to the corals, it's a mutualistic endosymbiosis, meaning that both partners are benefiting and one is living inside of the cells of another. These microalgae are actually dinoflagellates, which is a fancy term that means whirling flagella. So sometimes unicellular organisms have little tails, right? And so these guys have two of them, and it lets them sort of spin around if you look at them under a microscope. They are microscopic. You can't see them with the naked eye. And they're the same types of organisms that cause red tide, actually. So Karenia brevis is the dinoflagellate that causes red tide around here in the Tampa Bay region and elsewhere in the world. So I like to pretend that my research is relevant to that as well, but I have no idea if it really is because they're pretty distantly related when it comes down to it. But these dinoflagellates that live within corals or in association with corals and some other organisms, they're known as zooxanthellae, or Symbiodinium is one of the genera, or Symbiodiniaceae is the family. That's the group that we work with a lot. And I mentioned that they are photosynthetic. And so when you're trying to define mutualism and figure out how it works, you want to know the size of the partners in relation to each other. You want to know who's benefiting and by what means. And this is what we call a nutritional mutualism, okay? Because the algae can perform a type of metabolism that the host can't. It can harness energy from the sun and convert that into sugar, just like a plant could. I can't do that because I'm an animal. I don't think you can do it. It's an impressive trick, and I would probably trade having to eat every day for growing some algae in my skin, and I could just go outside and photosynthesize. We're much too big for that to work, but for corals, it can. And so the corals are actually putting a really heavy tax on these algae. So you think of the pie of energy that a alga produces, it might transfer 80% of that to the host, even more in some cases, less in others. So it costs a lot for these algae to be living with the host, but they get a lot out of it. The host is protecting them. It's keeping them in this nice habitat that's comfortable. It's supplying them with some of the nutrients that they need, some of the wastes that the corals produce or things that the algae need to perform their metabolism. And it's keeping them up again in the light, giving them access to photosynthesis, and the skeleton is even enhancing the light levels that those algae experience. These guys, when they're living on their own, they tend to be in the sediment. They're benthic. They live on the bottom, and they might not have many opportunities to encounter other algae. They're on their own, but once they're inside of a coral host, they can clone themselves like crazy. And every day, the coral has to expel a certain amount of them just to keep the population from overrunning the coral. So now you've got all of these clones of yourself getting out there into the world, and each one of them will now have a chance to reproduce. And that's one of the goals in life for many of these organisms is enhancing their reproduction. And so that's what the algae are getting out of it. And the host, of course, is getting energy to calcify. So in that way, they are both benefiting from each other. And overall, it's more beneficial to them to be together than it would be for them to be separated from each other. Of course, the amount of benefit can change depending on conditions. So, for example, at night, the light goes out and the algae are no longer photosynthesizing. Well, technically, at that point, they're just living off of the host and they're not providing anything to the host until the next day when the sun rises again, right? So if you just wanted to look at it at night, you could consider, hey, maybe that's kind of a parasitism, maybe. And what we're seeing now with climate change, a change in the environment, this mutualistic beneficial association under certain circumstances is becoming one that is not sustainable. That's where bleaching comes in. So if it's getting too warm, that algal photosynthesis breaks a little bit, and the nutrient cycling that's going on between the host and the symbiont breaks down a bit. And suddenly these partners who were tolerating each other are no longer doing so, right? And because of that communication breakdown, the end result is that the algae end up leaving the host. And in that scenario, you're basically leaving the host in a compromised position, right? It's no longer getting as much energy as it used to. We've got 2,000 calorie diets, right? What would happen if you had to live off of 300 calories a day instead of 2,000? You can do it for a little while, but eventually you get really weak, right? And that's what we see in bleached coral colonies. They can live for a couple of days, maybe a week or two. It depends on the severity of the bleaching, without their algae or without the really dense populations of algae. But they'll start to get sick. They're running low on energy. They're immunocompromised. Other diseases in the water that they could fight off before, they can't keep at bay anymore. And so unless the conditions improve, unless the temperatures come down and the algae can photosynthesize like normal again, those corals often die. And even if they do recover and their algal populations regrow and you get a working functional mutualism again, they might not reproduce that year, right? They had to spend so much energy trying to survive that they didn't get to spawn. And so that's really the concern long term with these coral populations. Not only are they dying because they bleach and die, but even the ones that bleach and recover, if we have these back-to-back-to-back bleaching events every year, they're never going to recover to the point where they're going to want to reproduce again. And then that just causes your population numbers to get even smaller. And so for some of these species, it feels like we're sort of spiraling the drain. There are so few colonies left. Their thermal tolerance is not great. Although the survivors have dealt with a lot of stress so far. So the ones that are sticking around are really hardy on some level. And then as the populations get spawner, you get fewer colonies spawning. They're farther apart from each other. There's less of a chance of their gametes mingling with each other. And the population just keeps getting smaller and smaller. This all feeds back in a negative loop.
Yeah, lots of information. Coral bleaching is a huge topic. You mentioned that some corals do recover from this, but okay, so my question is, if corals are so vulnerable when a bleaching event happens, what changes in the span of a few days that allows them to recover?
That's really going to depend on a lot of the circumstances of that particular bleaching event, right? Which coral species you're dealing with, which algae it hosts, what type of temperature extremes it's going through. So was this a quick hot water mass that came in and then left, but was really intense? Was it a lower, still elevated, but a lower temperature, but it stuck around for a really long time? Did the corals even have time to bleach? I'm sure I'll talk to you about this more, but we had a very, very bad bleaching event in 2023 here in Florida. And what happened is a warm water mass came and sat on top of the corals all summer. It came a month earlier than expected. It stuck around longer, and the heat was higher. We were out in the field doing work, and a lot of the corals that we were working with died within a couple of, well, we weren't out there every day, so I don't want to say it was just a couple of days, but we went out 1 week, they were alive, came back a week later or two, and they were dead. They were sloughing tissue. They were losing it. It was like they were boiling alive. They didn't even have a chance to bleach, right? It was just so hot that these particularly sensitive species were kind of melting. So what we're doing to the oceans right now is so extreme that even this bleaching, which we were thinking of as a sign of what climate change could bring, back when it was sort of first recognized as a problem in the 80s, in fact, that we had documentation of bleaching, and I'm sure it had happened before then occasionally, right? But we weren't out in the ocean checking it so often. In some ways, we're jumping past bleaching to now we're just cooking them if the temperature is extreme enough. So yeah, what dictates whether a coral can recover from a bleaching event? Part of that has to do with which algae it even has to begin with. And so this is another benefit, I guess, of some algae versus others. They can be more tolerant of that stress. I'd mentioned that their photosynthesis kind of breaks down. Well, some, it seems, they can persist longer. I think the host doesn't detect as much of a problem early on. If you want to quantify it, there are certain species of algae that can make a coral an extra 1 degree Celsius tolerant, so that the temperature could get 1 degree higher than all the other corals. They would all bleach, but this one would still be alive and kicking in a functional symbiosis for at least 1 more degree, which is actually a lot. So it's not, in that case, just a nutritional symbiosis, but it's also sort of a heat tolerance symbiosis. Or, yeah, I mean, it's all in support of maintaining the mutualism. But yeah, some species can recover quickly because they didn't lose as many symbionts, maybe, because the symbionts were a bit more heat tolerant. But usually what determines recovery is getting rid of that initial stress. I want to point out it's not just temperature that can drive stress. So elevated temperature, coral bleaching, especially in combination with high light. But you can get bleaching events at low temperatures. It gets really cold, corals will bleach. If you put a coral in the closet for a month and don't let it see sunlight, it will bleach. If you stress it with too much salinity or too many nutrients, it's this generalized sort of stress response, which kind of makes sense when you think about how integrated the host and the symbiont are. Basically, you disrupt one partner to an extreme degree, and they can no longer really function together properly, and then they end up parting ways.
So bleaching specifically would refer to when the algae goes away from the coral as a result of stressors. You mentioned that case where the bleaching didn't really happen, but the coral was still dying. Yeah.
Yeah, that was more than just a bleaching event. That was a thermal shock. That it wasn't just messing up the symbiosis, it was killing the coral. The technical definition of bleaching is a loss of pigmentation, which can happen through the loss of algae. It can also happen through a reduction in the chlorophyll that the algae produce, so loss of pigments. So technically you can bleach without losing symbionts if all of the symbionts produce less chlorophyll, if you want to get technical.
Yeah. Okay, great. Then I think that's a good place to transition into the work that you specifically do, which does look at the heat tolerance of corals, correct?
I try to.
Okay.
Yes. I've melted a lot of corals in my time. Yes.
The poor corals.
Yeah.
Thank you to Chopping Block Soaps for sponsoring this episode of Shellphone. This family-owned business makes my favorite soaps out there. They are dedicated to using high-quality, natural ingredients to create their handmade soaps and skincare products. One of my favorites is the Utah. This soap is made with shea and cocoa butter, coconut and olive oil, cedarwood and lavender essential oils, sage, beetroot, water, and lye. That's it. To browse Chopping Block Soap's selection of products, go to their website at choppingblocksoap.com and use the discount code Shellphone for 20% off your order.
The thing that's really dangerous and insidious about climate change is that it affects even healthy reefs, because we only have one ocean and the whole thing is warming. We're running this massive experiment where we're turning the temperature up and there's no pockets of safety. Whereas many of the other things that kill corals, we do have a little bit of control over. We can reduce the amount of waste that we put into the ocean. We can turn off sewage effluent. We can prevent people from overfishing, right? Those have sort of a bit more actionable goals, even though it can be very difficult to convince people to not fish somewhere, for example. And you have to have a whole marine park established and rules and consequences, etc. But the political will to reduce greenhouse gas emissions is a challenge, a major challenge of our time, right? And corals have been dying, and we've been talking about reducing greenhouse gas emissions for decades, and that hasn't really slowed things down, right? So I don't want people to come away with this thinking, man, there's nothing I can do to help corals. There are so many things you can do to help corals. And even when it comes to climate change, there are things you can do. But, you know, driving less, I don't want to say it's a drop in the bucket, or flying less. it does matter, and your behaviors influence other people. And if we overall as a society could become a little bit less reliant on these things, maybe that would move things a bit. But man, so much is done by industry, for example. If you really want to have the big impact, you got to look at that. You got to look at agriculture and how many cows are belching out methane. These are things that we individually can't control as easily. But supporting those causes and supporting the politicians who are emphasizing those causes is very important. And on top of that, you can do things like maybe don't buy the snapper when you're going out to eat, right? Or there's supposedly reef-safe sunblock, for example, that you can buy. Now, there's actually a lot of arguments about how safe reef-safe sunblock is, and it makes sense in some contexts and not others. I would argue always wear sunscreen. So it's not a lost cause, and that's something that's important, I think, for me to remember as a scientist, but especially for everyone to keep in mind. We talk doom and gloom a lot, and then when we talk optimism, people say that's a distraction, and you're just trying to make people feel good, and people should feel bad because that's the only way you're going to get action. I'm, I guess, an optimist, and I think there are things that we can do and should do, and that's why I do some of the research that I do.
Yeah, it's good to hold on to that optimism, and that's that's the reason why people want to make a change, is that they see something positive coming out of it.
And we have tangible examples of where coral restoration has actually worked very well, or prevented local extinctions, or even I can say, despite all of this death that we observed in 2023, there were colonies that survived. And I see that some colonies can hang in there, and that gives me hope as well. Those are the colonies that are surviving and reproducing and hopefully passing on that capacity to be tolerant to the next generation of corals. Our job really at this stage is to keep corals going through this very rough time for them, with the hope that we'll be able to mitigate climate change to some extent. And there's a light at the end of the tunnel. We'll get past it, and then conditions will go back to ways that corals, as we know them today, can potentially flourish again. I don't know if we're actually going to reach that. Coral reefs are definitely changing, and they will continue to change. And they might not be the same as they were before. And so that sometimes the idea with conservation or restoration is to keep things as they were before. I think we've moved beyond that. I think we've caused so many changes to these ecosystems that it's not going to be a pristine what it used to be. It might look different, but I think that we can embrace that change as long as the reefs are still providing the ecological goods and services that they give to both the environment and to humans. And so trying to keep that ecosystem going is something that's not only worth doing, but it's worth trying to remain optimistic about. Because we need more people in the future to keep doing this. And if the next generation goes, man, the corals, they're a lost cause, forget it, that would be terrible.
Yeah. Climate change seems like the kind of issue that you have to be more focused on adapting to and surviving as your environment is changing. But there is still the other side of it where hopefully we can not accelerate and keep making the climate change worse, and we can actually address the base of that issue. But adapting is still a huge part of it for corals and people and everything.
I think so. And I don't speak for every coral biologist. There's a lot of debate even within our own field about how best to go about messaging our discoveries and talking about the ways that we can effect change.
So you are a professor and a researcher. You've done research on corals and sea anemones, which we'll talk about as it relates to corals. Can you give us an introduction to that research and what kinds of research goes on in your labs?
Yeah, I think we do a lot of different things. And I think for the purpose of today, there are really two stories I should focus on. Okay. So one is some of our field work and our field heat stress tests. And then the other thing is the sea anemones, which we can save for the next part. So I mentioned that I was in the field during the 2023 bleaching event. What we were doing during that time was this experiment that we teamed up with our collaborators at Shed Aquarium, also the University of Southern California, Perry Institute of Marine Biology, and University of Miami. think that's everybody on this particular grant. But we got to use this boat from Shed Aquarium, and it's so cool. It's, I don't know, an 80-foot boat, and it's designed for research. They keep it in Miami, and then they bring it out, and we can drive it throughout the Caribbean. And what makes it unique is it has this live well system in the back that's just, you lift some planks up, and there's just a giant tub in the back, and you feed seawater in as you're driving the boat, right? And it keeps it aerated and flushed or whatever. And you can put entire big coral colonies back there if you want to. And so the premise of this particular project, we were originally going to work in the Bahamas, but COVID sort of got in the way. So we transitioned to Florida. What we wanted to do was twofold. One, we wanted to improve restoration. And so part of that involved building some more nurseries and moving coral colonies across those nurseries, helping to exchange them so that all of those unique individuals that were being held at one location were then spread out throughout other locations. I don't think I even mentioned what a coral nursery is yet, but we'll come back to that. The other goal was to stress test the corals and figure out what their heat tolerance was. And heat tolerance is sort of this wishy-washy term. What exactly do we mean by that? When you look at something and it's poisonous, okay, has a warning label on it, how did people find out that it was poisonous? Well, probably people would die from it. We'll start there. But how poisonous is it exactly? It depends on the dosage, right? And you'd have tests where you expose, say, mice, for example, to different concentrations of this toxin. And then you'd figure out the point at which maybe half of them died, because we all have different physiologies, right? We're all going to react to a poison a little bit different way. But you find that it's called the lethal dose 50, the point at which 50% of your population dies. That gives you some metric of lethality, right? We can do the same thing for heat. So what is the temperature at which 50% of your corals in the population maybe die? You could use death as a metric. We don't. We use something else. It's actually how well the symbionts are photosynthesizing. We have a non-invasive way of shining a light on a coral, and a certain amount of it's reflected back, and we can know how much of that light energy is going towards photosynthesis. And it declines as the coral gets stressed and as the algae in the coral gets stressed. So it's not perfect because it's a measurement of just the algae. But we look at, at what point are the algae reduced in their photosynthetic efficiency to 50% of their starting value. And we can do this because of a really cool rapid stress test assay that was developed, again, by people in the coral field. And it relies on the fact that you can break a coral apart into multiple different colonies or fragments of a single colony. And so we were going throughout the Keys. We were stopping at these coral nurseries. We were taking colonies up. We were breaking parts of them off. Some were being transported to the next coral nursery. The others were placed into these 8 different aquaria. And they were all very finely tuned aquaria where we can control with a computer exactly how warm it gets in each one. So we take one clonal colony, break it into 8 fragments, and put one fragment into each one of these little mini aquaria. And then we give them each different temperature stresses. So one stays at the control temperature. Another one goes up by 1 degree, another by 2 degrees, another by 3, all the way up to like 8 or 9 degrees above what they're usually exposed to. Those fragments, they don't just bleach, they start to slough tissue, right? But you get this whole gradient of response. And the whole time we're measuring their photosynthesis, and we can come up with an exact time stamp or temperature stamp. This is the 50% point, essentially. And for each colony, we do that. And so then we can compare them with a very specific number. Your heat tolerance is 36 degrees Celsius. Your heat tolerance is 34 degrees Celsius. The one that's 34 degrees is more sensitive, right? It can't handle higher temperatures. The one that's 36 is more tolerant. It's a better one to use if you're trying to plant a new population of corals that are going to be heat tolerant. You should focus your restoration on that particular coral rather than this one. And that's important information for these coral conservationists to have, because there's only so much money to go around to propagate so many corals. And they're really focused in the moment on ones that can be more heat tolerant. It's not the only stress that's out there, and you don't want to select only for heat tolerance entirely. But this was the idea, was to go out and survey all of these corals in Florida. We did over 250, I believe, measured them with this assay, and we could rank all those colonies, who's most heat tolerant and where are they from, and share that information with the nurseries. These nurseries are basically, they're like Christmas tree farms. And it again relies on breaking these corals apart into different fragments. You string them up on these PVC pipes and you suspend them in the water column, and it's just a way of propagating corals and making more biomass, because they grow rather quickly on these frames, and then you can take them off the frame and plant them, perhaps at a coral site that is not, or a reef that's not doing so well, or that, it did well in the past, and then a stress happened and it lost, 50% of its corals. But now that stress is gone. Well, now we can put some corals back there, and we think that they should do okay, unless another stress comes. In addition to fragmenting these colonies, moving them around, and identifying which ones are most heat tolerant, we were also sampling their genomes. So we've sequenced the entire genome of all 250 colonies. It's very shallow sequencing, which means it's less expensive and we don't have as much data necessarily, but it let us sample a lot of different colonies. And what we're doing now is called a genome-wide association study, where we have the genomes of all these different individuals and we have their heat stress tolerance levels. And we're looking for associations between different parts of their genome and heat tolerance. And we can do that by, grouping, these are our heat tolerant individuals, these are the ones that are sensitive. What's different? Much of their genome sequence is the same, but there are some regions that are different. What are those differences? Which gene regions do they fall in? This heat shock protein, that's really highly modified in the group that is heat tolerant. Maybe that's the key. It's just one contributor. There are many things that go into heat tolerance, but we're trying to find some genes that we can then use that information to perhaps enhance the next generation of corals. And I don't want to say we're there yet. We can't just insert a gene into a coral and make it more heat tolerant. But that's sort of the idea, that maybe down the line we can do that. And short of that, we can identify which individuals have those mutations and breed them and introduce them to populations that maybe don't have that mutation. So that, and a mutation in this case is a positive thing, right? It's making them more heat tolerant. So we can introduce that gene to populations that are maybe more heat sensitive, and in the next generation, they'll be more heat tolerant and sort of future-proofed as things get warmer. So that's one project that we did that I think tells you a little bit about what our group is focused on, and how we try to integrate conservation and all these nonprofit partners that are and universities that are running these coral nurseries and maintaining them and using them to actually on the ground restore corals right now in the Florida Keys. Of course, what's really sad is all these colonies that we sampled back in 2022, we went back in 2023 and they died. So we're learning about heat tolerance from a population that was devastated by an incredibly extreme heat. Unprecedented is a term that gets thrown around a lot, but it really was for this region. We have a way of measuring the amount of heat that a coral reef is experiencing above its stress tolerance levels. And usually you expect bleaching or paling at a unit of 4 on this scale, where you have a prediction for potential bleaching. You predict everybody to bleach at a unit of about 8. That summer, the unit went up to 22 in certain areas. Never been seen before. They had to add extra alert levels to NOAA's coral bleaching forecast system to accommodate the extreme temperature that was seen here and elsewhere around the world in this large global coral bleaching event. So these corals have survived a lot in the past. They can still be very informative for heat tolerance understanding, but they were just exposed to something no one could handle, essentially. And we lost basically all of this particular species, the Acropora cervicornis and Acropora palmata, 2 species. There are some patches here and there, but most of them south of the Middle Keys are gone, and they used to have healthy populations. There was a big effort in place while this was happening to actually save a lot of that genetic diversity. And so they're dead out on the reef, but fragments of them are alive in an aquarium. So I don't think any or relatively few of those particular corals were actually completely wiped out, but they're going to have to grow back from little nubbins. And that was like an emergency Noah's Ark operation to go out there to each of these nurseries and take some fragments and get them back to the lab, essentially the aquarium, and cool them off, because again, it happens very quickly, or in this case it did. So I told you I talked too much.
No, talking a lot here is great. There's so many super interesting points that are being brought up, and really informative points as well. I did have a question that I thought of while you were talking about all of that. Multiple questions, but the top question that I want to ask to follow up on that would be if there's a concern of a loss of genetic diversity when you're replanting or re-putting out certain individuals that you've decided, I guess, are more able to withstand these things, or a loss of genetic diversity when it's only those individuals surviving out on their own and not necessarily when it comes to researcher input.
We are absolutely concerned about a loss of diversity, definitely. And yeah, if we put all of our eggs into one coral basket and that one happens to be very susceptible to a disease, you don't want to plant just that one. You'd have an Irish potato famine issue, right? Everything would die suddenly. So we actually advocate for trying to maximize the diversity of the stock that you are maintaining in your coral nursery. We don't want you to get rid of the slow growers or the thermally sensitive individuals. But if, we want the information to be available to the people who are running the nursery, that might help them adjust their priorities. Like, okay, maybe this is something rather than growing an equal number of this coral and this coral, we'll pull back on this one a little bit. We'll still retain it for diversity, but we'll put an emphasis on, these heat tolerant species that we want to put, it depends on the project too. We want to put heat tolerant corals out someplace. Well, let's do these ones. If we want to propagate diversity, well, let's harvest a little bit of everybody in the nursery and put them out. So we definitely recognize that diversity is one of the biggest issues, and part of why we're advocating for moving corals around, which some people don't want to do, is just to improve diversity and to make sure that we have diverse corals in different places and that they're all spawning with each other and producing the next generation. Also, we're not just doing what I've described so far is asexual propagation of corals, but there's also sexual reproduction of corals as a restoration method. And you can go out there on a spawning night and plop a net over a coral and collect those gamete bundles that it releases, bring it back to the lab and mix certain individuals with each other and sort of control larval production. And then you can try to release those larvae back onto the reef, or you can settle them onto little tiles and then put those tiles back onto the reef once they've grown up for a little bit. You can artificially introduce new symbionts to those young settlers because when they're born, usually they don't have their symbionts. They have to acquire them from the environment. It's not true for all corals. Some transmit vertically from the parent, but a lot of them acquire it from the environment. So if we can provide them with something that we know has that extra heat tolerance, maybe that will help them survive into adulthood. So this whole sexual propagation component is meant to complement the asexual propagation because absolutely we're worried about diversity and we want to enhance diversity where we can. We want to create populations that again are self-sustaining, and one coral is not self-sustaining. I said they were hermaphrodites, but they don't really self-fertilize and produce viable offspring that way, and they don't get any new genetic material that way. You really need them to cross with other individuals. And again, that's one of the reasons why we are proponents of, I am at least, assisted gene flow, which is picking up colonies and moving them so that they can reproduce together. As I mentioned, one of the things that's happening is the populations are getting so low that they're getting cut off from each other. But these are clonal things. We can break half of a colony off and move it. Now you've got that same colony in two places. And you just doubled your chances of having it contribute to the gene pool. If we can move corals around and move some heat-tolerant corals in with some less heat-tolerant corals, that might, they're still being exposed to selection, right? It's not, we're always artificially manipulating things on some level, sure. But once you put two corals next to each other, they still have to spawn, they still have to reproduce with each other, and they're still producing billions of offspring. And again, most of them are going to die, but they could have reproduced together naturally. We're just sort of enhancing that by bringing them closer together, right? And or potentially moving them from one area to another one to take advantage of whatever local adaptation they had to that more high temperature site and bring that to a cooler site. There are concerns that by doing that, you're maybe moving microbes that you don't want interacting with each other. Or maybe it was so well adapted to that one site that by bringing other genes over to the new population, you're making that local population less well adjusted to where it's currently living. So those are reasonable concerns. And a lot of that comes from a traditional view of, the precautionary principle. We don't want to mess things up too much. We don't want the cure to be worse than the disease, right? And there are plenty of examples of humans and well-intentioned scientists going in and messing around with the environment, making it a lot worse. So I get where people are coming from if they object to the idea of assisted gene flow, for example, or of gene splicing corals and trying to create Franken corals that go out and, maybe spread throughout the world unintentionally. That's certainly not the goal. But I think for those who are concerned, the other thing you have to do is weigh it against the danger of doing nothing, right? And we saw with that 2023 incident, we're on the verge in the Florida Keys, at least, of local extinction of many of the species that have always been here, at least since modern scleractinian corals emerged. And we're on the verge of, if we lose them, we lose the habitat entirely. So would you rather have a disturbed and manipulated habitat that is functional or no habitat? And that's where I think and many people, to their credit, who have been critics of some of these ideas, as they've seen more and more corals die in certain locations, they've become more open to some of these ideas like genetic rescue and assisted gene flow. So I don't want to put anyone off, and I understand the precautionary approach, but it's dire stakes, and with these acroporid corals that are really the ones that were affected in this bleaching event, what we realized is that we waited too long to act for that species, and it's probably gone in the wild, right? And it won't survive without us intervening and helping it limp along in every year. But there are other species out there that aren't as far gone as the acroporids. There are still big contributors to reef structure, the orbicellid corals, for example, these boulder corals. They might not be so endangered yet, but now is the time to act with them, so we don't get to the same position we're in with the acroporid now. So that's my soapbox, is that I think we should be a little bit more proactive, even with some of the species that are doing well, and be less concerned about messing them up and more concerned about not doing anything. Was that your question? That was your question.
Yeah.
Yeah, okay.
You definitely got there.
Great.
There's a lot of weight that comes along with making decisions about conservation, and also a lot of weight that comes when things don't really go the way that you would hope for them to.
Yeah, we weren't hoping for all those corals to die. There's a lot of money went into propagating those corals, and it's been kind of a shift and a depressing shift in recognizing the limitations of what we can do. Yeah.
So you talked a little bit about studying corals in the field on the really cool boat setting.
Yes.
And that does sound super cool, yeah. But then another part of the work that you do is in the lab, and that is very different. It comes with all of its own challenges and goals and things like that. Can you talk about that difference between studying corals in the field and in the lab, and what your focus is when you're in the lab?
Absolutely. So you might have heard or gathered from my description so far that corals can be a little bit sensitive. We have the joke that if you look at a coral wrong, it dies. Maybe that's just because I do all these experiments where I heat stress them. They have lasted for millions of years, so they're hardy on their own, but they are sort of living at their thermal limits all the time, and you can push them over the edge quite easily. So corals are difficult to work with, let's put it that way, and it's great to go and study them in the field. That's where it's most relevant, but we have learned a lot about them by bringing corals back into the laboratory, into an aquarium setting, and doing our experiments there. One of the cool things with the whole field assay that I was talking about is we kind of bring the aquarium to the field in a way, and we can do those stress experiments right on the boat out in the water. And so we can look at a lot of different corals all at once. But a different way of approaching it is bringing the corals into the lab And that comes with sort of husbandry challenges, making sure you have a system with the right lighting and the right nutrients in the water and balance of pH and everything. And there are brilliant scientists out there who do awesome work in aquarium facilities and mesocosms and at field research stations and such. I am not very good at keeping corals alive. I acknowledge this, and that's okay. It's just not in the cards for me. And also, we can't go out into the field every day. So we have this workhorse that we maintain in the lab so that I have my students, they can do their experiments around the clock. It's these sea anemones, Exaptasia diaphana is technically the scientific term for it, or I'll probably just call it Aptasia. They're pale sea anemones. They're very common aquarium pests. They're very hard to kill. And that's what makes them great for what we do. That's one part. The other thing that's great about them is that they host the same type of symbionts that the corals do, those zooxanthellae, right? And on top of that, we can bleach them artificially in the lab. And we can reintroduce different algae to them in the lab. We can grow the algae outside of the host in the lab. So I've got incubators full of algal cultures in glass tubes, multiple different species. I think we have like 120 different individual strains. And part of what we do is we take these sea anemones and we bleach them, and we use a chemical for that. We don't expose them to a temperature stress just because it's less impactful on the host. So we use menthol. We soak them in menthol for a while, and they just start to eject their symbionts. We let them recover, and then we can basically squirt a bunch of new algae in there, and they take them up, and they repopulate with that new thing. And so we, again, in our lab, we're really interested in the interactions between hosts and symbionts. So this gives us a chance to look at, okay, if you have this one host, how does it perform with all of these different symbionts, some of which might be natural partners, some of which might be totally artificial? And are there any benefits to it in terms of heat tolerance, for example, or reproduction? We can make sea anemones spawn in the lab every month out of the year if we wanted to, because we can manipulate the light levels and how much they're fed. And if you do that the right way, you can spawn individual polyps, and they make larvae, and you can do experiments on them. Or you can just measure how many larvae they produce, for example, as some sort of response to if we were doing a temperature stress or a salinity stress again. So what really makes it a useful system is our ability to maintain both the host and the symbiont in this non-mutualistic state. And these sea anemones don't seem to be as dependent on their symbionts as corals are. So as long as we're feeding them some brine shrimp along the way, they're perfectly happy to grow without symbionts. They'll be fine. We keep them in the dark, and they're good to go until you expose them to light and new symbionts, then they'll take them up, and they're happy that way. Again, they're a lot harder to kill. They're very easy to propagate. We can clonally produce hundreds of them rather quickly, and we can mix and match them for experiments. And I can bring undergraduate students into the lab, or I can bring sea anemones to my undergraduate students in a classroom setting, and they can look at them under the microscope and do some of these experiments on their own. They're pretty simple and easy to do. And we've actually learned a lot through sort of the combination of classwork and research, bringing those together. They might not be the most complicated experimental designs, but they're still giving us some cool insights into how sea anemones work, which we can then try to apply to corals. And again, it's not a perfect match. So sea anemones and corals are divergent from each other. They've evolved separately, although they're pretty closely related to each other compared to other things. They do have the same symbionts, but the sea anemones don't calcify. So you want to study the impact of ocean acidification on calcification. You're not going to be able to do that with a sea anemone. There are obvious limitations there, and sometimes extrapolating from one association to another doesn't make sense. That's even true when we study corals. Just because one coral responds one way to a stress doesn't mean another wouldn't respond a different way, because they can be very different from each other as well. But still, on a fundamental molecular level, this is a very tractable system. We've got the genomes of both partners. We even have some gene editing skills for at least the sea anemone host. We can go in and manipulate things a bit. Well, people more talented than I can do that. But we can collaborate with them, and it all works out. But we wouldn't be able to do some of the things that we can do and have the type of replication that we can have with corals in the field, right? They're endangered. the permitting for them is getting harder and harder every year as more of them die, right? And that's one of the reasons we actually work with coral nurseries, is they have a lot of biomass that we can use in our experiments. But with sea anemones, there's no real restriction there. They're also fascinating on their own. Sea anemones you can find them on mangrove roots here in Florida. It's like a weird habitat that you don't really find corals thriving in. And there's a lot to learn about them too. But I think if we can get more of their natural history, that'll make it easier to extrapolate from what we're seeing in their molecular biology to that of corals. And we can really use them because there's still so much we don't know about cellular interactions between host and symbiont. I think what we pick up from sea anemones is still going to be relevant and at least guide questions in corals that we can then test in that system as well. And there have been plenty of experiments where we've tried something in a sea anemone and then tried it in a coral and found a lot of overlap in how things work. So there's good reason to use them as models.
My 2 follow-up questions to that whole topic, you've already answered. They were what the connection is between sea anemones and corals that makes them applicable to each other, and then also what would be the reasoning to choose whether to study something in the field versus in the lab, and you've touched on both of those. You did also mention that you have students who work in your lab with the sea anemones and other things. I'm curious if there are any students who have a different research question that they are able to use these resources that you have, the anemones that they're looking into, or if a lot of students are kind of looking at one central question, or
Absolutely. Okay.
if that's even relevant.
I think it is. I think it's good to understand how research universities work, right? I'm a professor. I have to teach, but I'm also expected to do research. And it's not every project where you can combine those things, right? I do have graduate students that can scuba dive, but I have others who can't, right? They just don't have the certification, or perhaps they're scared. I don't know. Everybody should learn how to scuba dive. It's awesome. And I don't want to knock my students who can't do it. They just haven't had a chance to do it yet. But it's an opportunity for students who maybe can't go into the field to do things, and particularly for my classes. I can't take 150 students out on a scuba diving trip with me, right? That wouldn't fly. It'd be difficult to coordinate. Although we do have some field courses, smaller field courses at USF for students that get them out into the reefs at Curacao and St. Vincent and the Grenadines. It's a really awesome program. But yeah, so sometimes there's a lot that goes into how you choose a research question. Often we try to find alignment between what the students are interested in and what my research goals might be. I have had students come in who want to do something completely different than what I knew how to do. I have someone who's working on bacteria right now, and he came in with a lot of experience in that area, and I was like, man, go for it. That's awesome. I don't know anything about bacteria, but they are other symbionts of corals, and they interact with the algal symbionts. So, and they can be indicators for reef health. So it still ties into conservation. So he's done this really cool work on looking at collecting eDNA around reefs and trying to use that to look at reef health, which is outside of my area. So I do like to let students guide things sometimes. Other times I've had an idea and man, I've got a grad student and that's the idea that they're going with because they thought it was cool. I think it's cool too, but it was my idea. So I think it's cool. But we often have sort of this hierarchy where there's me and then there's the grad students and there's undergraduate students who work with the grad students. And usually they're picking project. They spend their first semester maybe learning the ropes essentially. But then if they stick around with us, they get to pick their own project. It's usually something related to what the grad students are doing that maybe the grad students could really just use help with. Some of these things, it's a lot of anemones and you got to keep them all alive and you have to expose them to different conditions at different times. So it can be really helpful to have undergraduate students in the lab doing that work too. And, we include them on the publications that will eventually come out of work like that. I've had some undergraduate students spearhead their own stuff as well. We had someone do a microplastics project. She looked at whether symbiont uptake was affected by when the host is exposed to microplastics, which looks like it is. What a shocker, right? Mm. But if you stress out a coral host with plastic in its gut, it doesn't take up as many symbionts as it would otherwise. That was a totally undergrad-run project. But then a lot of our projects sort of overlap, or we have students helping each other out as well. I think everybody is they wouldn't come to this lab. There are other options out there. They wouldn't come to this lab if they weren't united in their desire to learn more about how technically cnidarian dinoflagellate associations, but coral algal associations work, and how we might be able to apply some of that in conservation practice. So I think everybody's really happy to help each other out. And I've been incredibly lucky with all of the students and staff who have worked with me. They've been awesome and very collaborative. So I hope that continues.
Yeah. Awesome. Well, I think we've covered a lot of ground, and that could be a good place to wrap things up. Is there anything else that you want to make sure that we include in the episode?
No.
Okay.
No, I think we're good.
We've gone over
I talked a lot.
So
Yes.
I think if for anyone interested in any of the topics that we've talked about, there's a lot more to explore, and you could definitely go and find resources that go more in depth or relate to anything that we've talked about. But yeah, awesome. Thank you.
Thank you so much for having me on here. And thank you to everybody who's listening for paying attention this long. and hopefully you'll come away with this with feeling inspired that you can help the corals too. Just by talking about them is a great thing to keep them in people's minds. Not everybody gets to go out onto a reef. I'm well aware of that. But man, there's so many great documentaries and videos just on YouTube and whatever that you can look at and learn a lot more. So I encourage you to do that.
Is there anywhere that if people wanted to learn more about your work that they should go to?
My work specifically, I have a website. It's thelifeaquatic.net. It's got a list of our publications. They're probably pretty jargony and science heavy, but there are a couple on there that are from, we wrote something for a coral magazine that's sort of an overview. I'm actually, most of what I work on is the coral symbionts side of things, their diversity, their taxonomy, their genomics. So a lot of it is focused on that. We've also got a book chapter, easily accessible for people who are getting into the field. It might still be a bit heavy, but those are places that you could potentially start. But NOAA has a great website. If you look up the Coral Restoration Consortium that started off as part of NOAA, I'm not even sure if it's still part of NOAA at this moment, but it's an awesome organization. They produce a lot of cool things, webinars, websites, species checklists. You can learn a lot about different groups by looking at the resources on their website as well. And then there's the International Coral Reef Society, which also produces a lot of infographics and information that can be used in a classroom setting or what have you. So you're right, there's a lot of resources out there for learning about and teaching about coral reefs and how you can get involved in helping them. I guess one other website I'll point you to is the Coral Restoration Foundation.org maybe, which is a big coral nursery operation, and they have a pretty impressive website with information about what they do and how you can even go there and help them and go diving with them and help clean a nursery.
And if you want to hear more about Breach the Surfaces projects or suggest a guest for Shellphone, you can find us at our website, which is breachthesurface.org, or on Instagram at Shellphone Podcast or at Breach the Surface. And thanks for listening to this episode. If you're watching on YouTube, make sure to subscribe to the channel so that you can keep up to date with future episodes. You can also subscribe on Spotify, Apple Podcasts, or wherever you're listening to this. See you next time. The end.
Yay.